on diversified topics. The diversified topics, which I said, which I'm trying to bring out here is, we spoke about, or we heard about wastewater, we heard about freshwater, we heard about lakes rejuvenation, the pond rejuvenation, pond conservation, and many more things like that. And very technical talks also were a part of this water talks, wherein we heard from something on robots that can help in assessing the condition of the pipelines in water and wastewater supply systems. We had up to 15th talks. We had several aspects that were understood by many people. A lot of interactions happened. And we had Sangeet Adhikari also, who spoke about wastewater epidemiology in the 14th edition. So this is what we have done till now. And of course, from all these talks, what we learned, how we are going to take forward, we are collecting it all in a form of a coffee table book. And hopefully we'll be releasing it soon in the next 15 days or so, on how many people have contributed to this, what is the talks that they have given, what they have inspired us on many topics of interest to the society. So we'll be collecting all your inputs from the talks that you deliver with us, and we will make it a point to enrich the stakeholders more and more on several topics like this. So with all due respects to your subject and your professionalism and expertise in this subject, we request you to give us that same kind of interaction that we wanted, and we would like to learn from you more and more on this subject. We will have in between some questions if possible. We will try and moderate it as smoothly as possible. We will take questions in the chat box, and we will highlight it to you for your answers. So Dr. Lauren, over to you now. I don't want to stop you more in introducing to the participants. Jennifer, would you like to say something about Dr. Lauren before she starts?
Yeah, sure, sir. Yeah,
please.
Sorry,
Lauren, we are delighted to have here. And am I audible?
Yeah, you are audible.
I logged in.
Some noise is coming.
Please go ahead.
So, an environmental engineer and assistant professor at Rice University. Since the outbreak of COVID-19, many wastewater-based epidemiologists have been working along with frontline workers to report the spread of viruses and to alert the public health departments. And so Dr. Lauren and her team have been also working on wastewater-based epidemiology along with the Houston Health Department, providing early warning signals to the public health department so that many lives could be saved before the spread of the virus. And speaking about her, she is also named a new engineer to watch by the Water Environment Federation. And as a dynamic engineer, she has won many awards, such as the Gulf Coast Early Career Research Award, Johnson & Johnson Women in STEM 2D Award. And she has received many scholarships, Fulbright Fellowship, National Science Foundation, Graduate Fellowship, Michigan Water Environment Association, John P. Hennessy Fellowship. And her work mainly revolves around sustainable biological wastewater processes. And we are here to hear about your WB journey, and kindly share your experiences with us. Over to you.
Great. Thank you so much for that very kind introduction, and for the and for the opportunity to speak in this series of talks. It's a real honor, and I hope I can share some of the stuff that we've been doing and have a discussion with you all on this topic. So I'm first going to share my screen. Okay, and let me know if you.
Yeah, we are able to see your screen.
Great, perfect. Okay, so as Jennifer mentioned, I'm an assistant professor at Rice University in Houston, Texas. I've been working closely with the health department, as well as many other universities and institutes across Houston and outside of Houston, to establish really one of the most expansive citywide wastewater monitoring programs in the US. just make sure. There we go. Of course, collaboration is critical to the success of this program. And this team has been led by Dr. Hopkins, who is the Chief Environmental Scientist at the Houston Health Department. She also has a joint appointment in the Department of Statistics at Rice. And Dr. Kathy Enzer and myself, we think of it as divided into kind of three main branches: the wastewater engineering piece, the analytics piece, and the implementation and leadership. And there's been many different folks across many different institutions that are critical to this program's success. And I just list all their names here, and you can see that they're from all different departments. And so they have to work together to make this work. So I know you guys have already had a talk on wastewater surveillance, so a little bit of this may be repetitive, but just as an overview, we know that infected persons, but whether they're symptomatic or asymptomatic, will shed virus in their stool. And this has turned out to be a very reliable indicator of infection. And because the virus is shed in the stool, it ends up in the wastewater. And that wastewater thus represents a pooled sample of all the individuals that have contributed to that waste stream. And that wastewater travels through the sewer system to a wastewater treatment plant, and we can grab a sample of that untreated wastewater and measure the amount of virus or viral particles in that sample to get a sense of the infection burden in the community. And we can also do this at different scales. So we can do it where we grab a sample at the wastewater treatment plant, and that's capturing all the individuals in that sewer shed that contribute to it. But we also can go upstream into the sewer shed and sample at lift stations or manholes or even individual facilities. And there's been a lot of work where people have even shown that this is a really valuable approach at the facility level, where you can basically assay the presence of an infection in a facility like a dorm or a nursing home or a jail. And people are extremely excited about this approach all over the world. And that's because it has some really critical advantages as a additional piece of information to monitor disease in communities. Oh, okay. So some of those advantages are, first, like I already mentioned, you can detect both symptomatic and asymptomatic cases, whereas clinical testing is really biased towards symptomatic individuals. You're probably not going to go get tested unless you're symptomatic for like a PCR nasal swab. It's very resource efficient because it's this pooled sample, and it doesn't require people to opt in because everybody uses the restroom. It does not depend on individual testing and reporting. Again, and the next one does not depend on health-seeking habits. So a lot of individuals who are getting tested, clinically tested, were symptomatic. There's those who had access to testing, those who had time to go get a test, those who had, flexible jobs where if they got a positive test, they could actually take time off or work from home. And so there wasn't a huge incentive for people to get tested if it meant, for example, that they had to take time off work. And now, at least here in the US, people are not really even going to get tested anymore. Everyone's doing the home tests. So when you do a home test, that doesn't get reported up to the health department, and that information is therefore not like aggregated at a population level. But wastewater, people haven't drastically changed their bathroom using behavior. So it's this nice metric that we can use that's independent of all these health-seeking habits. And then lots of studies have shown that wastewater data can actually trend. Oh, I think someone has their microphone on.
Jennifer, can you please mute all the people?
Yes, sir, we'll do that.
Kindly mute all the people, and then Lauren can unmute herself, and then.
Suddenly someone has come up. We had put all of them on mute.
Okay, all right.
Go ahead, Dr. Lauren.
Yeah. Okay. And of course, we have to keep in, I was saying lots of studies have shown now that wastewater can actually trend before clinical data, right? So you can actually get an early warning signal by looking at the viral levels and the changes in viral levels in the wastewater And lots of people have also showed you can detect variants circulating in communities before clinical testing makes that information available. And we also saw that in Houston, and I'll talk a little bit more about that in a few slides. Of course, we have to keep in mind that it requires that you have a sewered wastewater system where you actually could collect, untreated wastewater. And of course, it's not perfectly sensitive. So a negative wastewater result doesn't necessarily mean there's no SARS-CoV-2 infections in your community. And there's lots of different factors that will impact that sensitivity, some of which I'll bring up in this talk. And of course, this is not the first application of wastewater-based epidemiology or wastewater surveillance for diseases. Long before SARS-CoV-2, this was being implemented to look for other viruses like poliovirus, in addition to some other enteric viruses. And it was used, for example, to detect if a virus was like poliovirus was circulating in an unvaccinated population to trigger a vaccination campaign. It's also been used to provide early warning of outbreaks, to understand which viral genotypes are circulating in a population. SARS-CoV-2 was really the first demonstration at a mass scale of wastewater-based epidemiology for a respiratory virus. Because of course, the type of infection is going to impact the signal that you see in the wastewater. So this was really the first time we saw, okay, we can use this for a respiratory virus. And it's been embraced and applied across the world. In the US specifically, it's now being supported by the CDC, so at the federal level, and they're actually trying to fund health departments all across the country to implement wastewater surveillance in all 50 states. So they're funding health departments all across the country to do this work. It's made news headlines to the point where probably very few people had heard of wastewater disease surveillance before the pandemic. But now if you ask someone, oh, have you heard of wastewater surveillance? Chances are that some people have heard about it in the news at some point. And so there's a lot of excitement about using this approach and a lot of case studies now where we can see an actual demonstration of the value of this approach. If you haven't seen this website, this is a map that's it's called COVID Poops 19. It's they aggregate all of the groups doing wastewater surveillance all across the world. And you can see there's over 3,000 sites that have been monitored all across the world, 140 public facing dashboards, almost 300 universities and across 64 countries. And it's amazing to be part of such a fast-moving community of researchers, scientists, public health, and utilities, all working on this common goal of implementing SARS-CoV-2 wastewater surveillance across and amidst this pandemic. I zoomed into India. There are a number of groups here in India doing wastewater surveillance. I'm not sure if your group is on here yet, but it should be, and it's if not, you can reach out to them and they'll put you on the map. But you can actually click and see, is there a dashboard where they report out results in real time? Where are they doing this? Which groups are working on it? Are there any papers associated with it? So it's a great site to kind of poke around and see what's going on near to you and also all across the world. So now I'm going to switch into talking about the wastewater surveillance system here in Houston, Texas. So Houston is the 4th largest city in the US. It's unique for a number of reasons, and I'll show you in just a minute why it's kind of a unique system. Here is a timeline of the COVID-19 pandemic and wastewater surveillance system in Houston. In blue is the timeline on the top of the wastewater surveillance program, and in orange are the big COVID-19 pandemic moments. So the city first shut down because of the pandemic in March of 2020. We started collecting our first samples at the very end of April and beginning of May. And it really took us between May and July of 2020 to reliably generate data for all of the wastewater treatment plants across Houston. And we have 39 wastewater treatment plants in Houston. And then after July, the system has just expanded to numerous other sites. And I'll describe some more of these moments as I go through this talk. So here's a map that shows where we sample the wastewater in Houston. As I mentioned, we have 39 wastewater treatment plants. That's somewhat unique, at least in the US. Most major cities and metropolitan areas have just a handful of very large centralized wastewater treatment facilities. Houston's unique in that it has lots of smaller wastewater treatment systems, but it also has some massive ones too. So we have one wastewater treatment plant that serves over 500,000 people, and that's that pink one in the middle, 69th Street. And then we have all these smaller ones, some of which serve less than 1,000 people. And in total, it serves about 2.5 million people, a little bit under that, and 670 square miles. I should have converted that to kilometers, but it's a really large geographic area. And we've expanded, in addition to these 39 treatment plants, to monitoring schools. So we monitor 50 schools, 23 congregate living facilities, which include nursing homes, a jail, homeless shelters. And then we also have about 60 sites at lift stations. So these are sub-sewer shed locations where we can break down some of these very large sewer sheds and get more resolved community information. And this is a logistical lift because there's so many sites over such a large geographic area. But this is an overview of kind of what happens each week. So 24-hour composite samples, meaning they're not just grab samples of wastewater. There's actually auto samplers installed at each of those locations I listed on the last screen. They collect one sample every hour to create this 24-hour composite sample. Those samples are collected and delivered to my lab at Rice University and also delivered to the Houston Health Department lab, where we each independently measure the concentration of virus in the wastewater samples. We also enrich and sequence the viral genomes out of these samples, and we use that sequencing data to screen for variants of concern. That data is input into a statistical model that identifies areas of concern based on changes in viral trends over week. And that information is reported out to health department leadership, and they use it in addition alongside other markers and clinical data to prioritize areas for intervention. So I really run the laboratory side for Rice, and we spent a lot of time developing methods associated with measuring this virus in wastewater. And this is just showing an overview of kind of the two different pipelines. So the top one is, I'll first talk about quantification, where we just measure the amount of virus in the sample. And then the bottom pipeline is the sequencing pipeline that we use to screen for variants of concern. And I'll talk about that next. So for quantification, it's very similar to how we measure virus in a nasal swab. We look for the genetic material of the virus. The difference between a nasal swab sample or a saliva sample and a wastewater sample is the virus is going to be present at very dilute concentrations, and it's going to be in this matrix of all the other junk that's in wastewater. And that other stuff in wastewater can impact your ability to detect these trace levels of genomic material of the virus. And so you have to do a step up front where you concentrate the virus in your wastewater samples. So there's three main steps associated with how we measure this. First step is concentration. Next, we purify the RNA or the genomes of these viral particles. And then the third step is we actually quantify very small fragments of the genomes. So we have evaluated every single step of this process and the choices at each one. I'm not going to go into a lot of detail in each one, but we compared 5 different concentration methods in terms of sensitivity, reproducibility, cost, and throughput. We compared 3 different extraction platforms, and then we also compared quantification platforms to see which one would be most sensitive and most reproducible. I'll give one example for the last, where we compared different quantification platforms. We compared something called reverse transcription quantitative PCR to digital droplet PCR. Both of these are basically targeting a very small region of the genome and trying to quantify the number of copies of that fragment of the genome. Digital droplet PCR is a newer technology, but people are really excited about it, especially for environmental monitoring, because it's very sensitive. It does absolute quantification, meaning you don't need a standard curve. And it's less impacted by inhibitors that are present in wastewater. So here I'm showing, we took the exact same extracts and we quantified the N1 gene of the SARS-CoV-2 by droplet digital PCR and compared it to qPCR or RT-qPCR. And you can see that for both the N1 and N2 genes, we determine higher concentrations when we use digital droplet PCR over qPCR. We also looked at, okay, do these results give us similar findings? And we see, yes, they're highly correlated with each other. So whether you're using qPCR or ddPCR, you're going to get similar information, which is great, right? That means even if you don't have a ddPCR machine or a dPCR machine, you can still use qPCR and get important information. But digital PCR is going to give you potentially more sensitive detection. So we went with digital PCR for our quantification, and we kind of did this for every single step. And so what we do is we take 50 mils of a sample, we add some salt to it, and then we filter it through an electronegative filter. We then take that filter, which captures the viral particles on it, and then we bead it to release the genomes. And then we extract that viral RNA using a system called Chemagic 360. We pick this because it's very high throughput. We can do 96 well plates. And then we use digital droplet PCR for quantification. And we published a methods paper on this, and I'm happy to share that kind of goes through how we came to these decisions. So now I'll start to show some of the data that we've generated. This is kind of an aggregate picture of all the data we have to date. So in the green line, I'm showing the viral load, which is the concentration of the virus times the flow rate of wastewater. It's relative to a benchmark level instead of, and we decided to do that because that was better understood by health department leadership than saying, you know, this many copies of virus per liter of wastewater. That was a hard to conceptualize idea, just because people don't really think about concentrations of virus. And then the gray bar around it is the 95% confidence interval. And then the positivity rate, the clinical positivity rate, is shown in blue. So each hatch mark is an individual day, and then the line is the 14-day moving average. And what you can see is these two data sets are highly correlated with one another, and they move with each other. And this has allowed us actually to, because they're so closely correlated with one another, actually use the wastewater data to forecast the clinical positivity rate. And we've looked at this not just aggregated across Houston, but at each of the individual wastewater treatment plants as well. So here I'm just showing 6 different treatment plants out of the 39 that vary from the largest treatment plant to some of the very smallest. And of course, we see with the large ones, you know, it's very close, it mimics it very closely. The positivity rate data is much tighter, of course, because it's a much larger population. And then when you get to these very small treatment plants, the clinical data is extremely sparse, right? Because this is only maybe 1,000 people or so, and not that many people are getting tested out of a population of 1,000 each week. And so your clinical data becomes, you know, all over the place. And so the wastewater data really represents kind of a more reliable source of information on infection dynamics in those communities.
If I may ask a question on the previous slide. So as we keep going, we are seeing these waves, and these waves are, the width of these waves is decreasing as time progresses. The early time, the waves themselves have many sort of multiple peaks in this. So how do we understand these related to the occurrence of specific kinds of strands, or how do we understand this?
Yeah, so definitely. So these, I mean, we can look at these waves and I can tell you what each one of them are. And I definitely think that you're correct. The waves are getting much shorter in duration. So this is our first peak. So this was when, you know, COVID first came through Houston. This is Alpha, this is Delta, and this is Omicron. So you can see each wave here very clearly in terms of how it kind of spread across the city. And you can see these waves were much broad. The alpha wave was significantly wider than the delta wave, which was significantly wider than the Omicron wave. And so I don't know what the next wave is going to look like. Actually, we're kind of experiencing an uptick with BA.2, but it's kind of yet to be seen how big this wave is going to be in terms of the magnitude of the peak and whether or not we're going to see it shoot down as quickly. So many things are changing right at the same time. The virus is evolving. More people, we're probably getting closer to herd immunity at this point, right? With everyone having been infected or vaccinated. And so it's hard to know what this next wave is going to look like.
Why is the, very nice. Why is the rise is faster and the decay is slower? There's some kinetics of these.
For Omicron?
Yeah.
Yeah, I think that's something that people, all over the world have been thinking about it. We don't really know. Omicron shot up, right? Everybody got sick, but it was so quickly, it was so fast that it plummeted after that we had that huge peak. And I don't know if it was just, okay, everyone got infected rapidly and there was no more individuals to infect, and so you kind of had this
So at the bottom, when you start seeing a rise, how does it correspond to do we know, let us say this is about 100 people infected over this million people, then you see as a rise? What is that first rise? How many people are infected?
I don't know if I know the answer to that question. That's a good question. You're saying how many individuals correspond to when you see a change in the inflection?
Right.
So I think it has changed for each wave, first of all. So I don't think it's necessarily the same for Alpha versus Delta versus Omicron. But I think that's going to be a very important thing. And we think about what is going to trigger an emergency response, right? It has to be maybe a certain change in wastewater, which corresponds to a certain change in, for example, hospitalizations or cases. I actually I think I have a slide that might be able to indirectly answer that in just two more slides. Yeah. But so we also did some modeling where we looked at the decay of the waste. We looked at decay rates of SARS-CoV-2 reported in the literature, and we measured it ourselves in the lab. And we tried to model the decay of the virus across our sewer system. And so here I'm just showing our largest sewer shed versus one of our smallest. And we're looking at different decay rates. So this is one value that's been reported in the literature, and this is another. They're drastically different from each other, which kind of just suggests there's really a need to figure out what impacts the decay of virus in wastewater, because there's such little data on this. And these are just showing different decay rates. This is the same sewer shed with the same travel times and hydraulics, but it's saying, okay, this is the slowest decay rate reported in literature, and this is the fastest. Decay will matter quite a bit more in these very large sewer sheds, and it's very decay rate dependent, and which is also temperature dependent. Whereas in these smaller sewer sheds, we really don't see very much loss of the virus across the sewer shed as it's traveling. But this means that it's important to compare, sewer shed to sewer shed, sewer shed to itself over time versus sewer shed to sewer shed, because you can have these impacts of decay of virus across the sewer shed that could kind of impact the concentrations that you're measuring. And so this is just an example of some of the information that we're reporting out to the health department leadership each week. This is a heat map with all of the sites on the y-axis. This is the dates on the x-axis. And again, we're showing it a comparison of the viral load to a benchmark level. The level we picked is the July 6th level, which is really the peak of the first surge of COVID in Houston. We picked this date because we had reliable data for all the wastewater treatment plants, and because it was a level that was really understood by leaders. And again, you can see these waves, Alpha, Delta, Omicron, coming across Houston. We also have thought about whether we can relate the viral load to the number of cases, so not positivity rate, but actual positive cases. And so this is for, I think, a 3 or 4 month period where each triangle represents a wastewater treatment plant. On the x-axis is the total viral load, so the total amount of virus that came into that treatment plant. And then on the y-axis is the total number of positive cases in that wastewater treatment plant sewer shed. So you see this very strong correlation between total viral load and total cases, suggesting that we may be able to use viral load to predict case numbers. And this is the slide I was talking about that might provide some indication of how this information could be used to relate to hospitalization numbers or set threshold levels that would trigger an emergency response, especially as we kind of transition out of pandemic mode into more endemic mode. So historically, we've been comparing wastewater viral loads to positivity rate, but as I mentioned earlier, fewer and fewer people are getting tested. So now we're trying to relate it to other syndromic surveillance. So things like emergency department visits and hospitalizations or percent ICU beds in use with COVID. And so you can kind of look at this and compare it to the wastewater viral load, which is shown in the blue. And this change in the wastewater viral load that corresponds to this, actually, usually it's a trailing change in hospitalizations or ICU beds used for COVID. And so we can try and use this information to say, okay, what is the threshold and change of wastewater that indicates, okay, we need to start thinking about there's going to be a surge in a hospital. And this is just showing, again, the 2 different waves, Delta versus Omicron. And of course, like I mentioned, there's a very different pattern in terms of wastewater viral load versus hospitalizations for these different waves. We see a much higher viral load, but a lower hospitalization rate for Omicron than we did for Delta. Okay, so now I'm going to shift over to talking about how we detect variants of concern using sequencing of the viral genomes. This has been collaborative with Dr. Todd Tregan in the Department of Computer Science. And we developed our own custom bioinformatics pipeline to do this work because it's challenging to detect variants of concern in these pooled mixed wastewater samples. And it's challenging because wastewater is this mixture of all the variants and strains that are circulating in a community, which means that all the mutations that are, that occur in a variant, that comprise a variant, are going to be present at variable frequencies depending on the abundance of these different variants in this community. So in clinical data analysis, clinical sequencing data, what's typically done is something called assembly, where you try and take all these individual sequencing reads and stitch them together to come up with a consensus genome. And that assembly process really averages out a lot of the information in your sample. And in wastewater, that would actually result in the loss of very important diversity information in your sample, especially if you're trying to detect the emergence of a variant of concern in your community early on, where it's present at very low abundance. You really cannot use assembly because it will just mask all of that emerging information that's showing up at low levels in your wastewater sample. You also can't use an approach where you just look for what we call characteristic mutations. So these are the mutations that basically give these new variants their characteristics, like enhanced transmissibility or enhanced infectivity or antibody evasion. And that's because these characteristic mutations, turns out, are shared by many different lineages. And so if you detect it, you don't necessarily know that it's one variant over another. You could try to look for a set of characteristic mutations and say, okay, if they're all present, I know it's this variant over this variant. But the problem with that approach is, again, if you're trying to do this for early warning signal and early detection, for all of your characteristic mutations to be detected, typically that variant has to be pretty abundant in your sample. And by that point, wastewater is not really that useful anymore. We're trying to use this to get early detection in our communities, not, okay, let's describe what we know is already present in our community. So we developed this approach that's called Quasi-Unique Mutations. Very simply, we define a quasi-unique mutation as a mutation that's present in 50% or more of the genomes of this variant of interest, but not present in the majority of other variants. So we can look at all available sequencing data for these variants, identify these quasi-unique mutations, and then screen for them in the samples. This is just a heat map of what this data looks like. So I'll just walk through how we interpret this heat map. The y-axis is all the different sites, and the x-axis is all the different quasi-unique mutations. So each one of these is a single nucleotide polymorphism in blue, and then a deletion in red. So if it's white, it means there was no data mapped that position, meaning we just didn't have any reads to call whether the mutation was present or not. And so one important takeaway is there's a lot of white on this, right? This is a sparse data set. You're not able to necessarily recover complete genomes of individual variants using this approach. Next, the blue means, okay, reads mapped that position, but there was no support for the mutation. And then yellow through red means a mutation was detected for that variant of interest, and it was present at different strengths or different allele frequencies. And so what you can see here is that, you have a collection of samples where you'll detect no support for mutations present in a variant of concern, but then you'll have a few sites that light up that do indicate the presence of this variant. And what we've seen over time, because we've been doing this, even before the Delta variant, is that just the presence of a handful of these quasi-unique mutations, whether that be a couple in an individual site or a few across several sites, is indicative of the emergence of a variant of interest in a community. And so we use this approach to basically screen the wastewater, starting from Delta through Omicron, and we were able to detect quasi-unique mutations for Omicron at 6 wastewater treatment plants before any known clinical cases. And so this is moving kind of fast. I don't know if I can pause it, but we first detected Omicron at 6 wastewater treatment plants on December 2nd. And then by the next week, it was basically at almost every single wastewater treatment plant. And this was a nice demonstration of how we can use wastewater surveillance to detect stuff before known clinical cases. Okay, so now I'll talk very briefly about manhole sampling. So this is not at the 39 wastewater treatment plants, but it's point surveillance at schools, the jails, nursing homes. This is just a picture of some of these auto samplers installed in the manholes. They either hang in the manholes or they sit outside of the manholes, and the tubing runs into the sewer system. These are reported out as we get this information. And this is just an example of the school data that we've generated. So this is all the 50 schools we've monitored as an example of a report that might be sent out each week. And this is dates. And we report out, okay, because we don't have any flow information for these schools, we report out whether or not it was detected or not. So negative is green, inconclusive is yellow, meaning it was like one sample was positive, maybe one was negative, and then red are positive and high positive. And this information is relayed to the schools, and the health department works directly with them to take action. And this is just an example of some of the actions that have been taken using this information, assessing past COVID history, current COVID status, whether or not there's a current outbreak, providing outreach to the school point of contact for health reporting. They've used this information to direct vaccinations and vaccination clinics. So these are just two examples of where they've hosted successful vaccine clinics, both for COVID, but also for influenza at elementary schools, using some of the information from the wastewater monitoring to decide where to do this. And then health department also hosted a school nurse webinar. So this information is communicated to the school nurses. And so our health department authority, Dr. Peirce, did a webinar where he walked through, okay, this is how you interpret these results. This is how you use this information. And then they did a survey and asked them, does having this information help plan and implement strategies to reduce the risk of COVID-19 infection on my campus? And most participants, these are the school nurses, answered agree or strongly agree, which was really validating to hear, because it's nice to know this data that we're generating on a weekly basis is actually being used for decision-making by the people that we're sending it back to. And it doesn't just go to school nurses. So these reports also get sent to health department leadership and even the mayor of Houston. It's sent to outbreak teams. It's sent to vaccination clinic teams, school nurses, and then we also make the data publicly available on a dashboard. So here is a screenshot of our dashboard, and you can check it out if you're interested at this website. And here we show every week the results in real time for all of the wastewater treatment plants. Most people don't know which wastewater treatment plant they contribute to. So you can actually search by your zip code instead of your treatment plant, and it'll tell you your viral load and whether or not it's increasing, decreasing, or plateauing. And so we've seen a lot of interest in this dashboard. Increasingly, people look at the wastewater and even potentially trust it more than they do the clinical data, because so few people are going to get tested anymore. But the wastewater is this reliable indicator that kind of shows whether or not we're seeing changes in infection dynamics across our city. Okay, so I'll spend the last portion of this talk talking about what's next for disease surveillance. We built this entire system for a single viral target, right? For one virus, SARS-CoV-2. But, there's tons of other stuff in wastewater, other diseases that could be monitored, other chemicals that could be indicators of health or disease or addiction or abuse. And so there's just so much of potential for using wastewater to look for other public health targets. So I'll talk about a couple of other disease targets that we've begun monitoring for. So influenza is one, and then we're also working on antimicrobial resistance. And then we're also exploring non-targeted methods. So we are not looking for a specific virus, but we're actually just sequencing all the genetic material present in the wastewater to see what we can detect and learn about discovering viruses in different communities and deciding whether we can use that information to design targeted assays that we can measure and quantify. This is just a table of some of the additional targets that we're working on. And they range from, other respiratory viruses like influenza and RSV to foodborne illnesses like norovirus, salmonella, campylobacter. And then we're also starting to look for antibiotic resistance genes, and particularly ones that have a lot of clinical relevance, like, for example, colistin resistance, because colistin is one of these last resort antibiotics. So we're starting to implement and develop methods for all of these different targets. We have a pilot program, or I don't know if it's a pilot anymore, an influenza monitoring program at the schools already. So we take the same samples that we monitor for SARS-CoV-2 and we assay them for influenza A and B. For a long time, we're not detecting anything. We were like, uh-oh, our method's good. We had checked them. We weren't seeing any influenza. It turns out there really wasn't any influenza in Houston at all for a long time, likely because SARS-CoV-2 is the dominant respiratory virus circulating in Houston, but we have seen an increase in influenza across Houston. And with that, we have started to see some positive detections of influenza or flu in these schools. And so this information, in addition to the COVID information, is reported out to the school nurses, and they can use this to kind of try and identify, sick individuals, make sure they stay home, and report out any known cases. We are also exploring untargeted methods. And so, there's pros and cons of doing kind of target-based detection, which is what we've done for SARS-CoV-2 and influenza, compared to sequencing-based screening. With sequencing, you can monitor, you don't have to know what you're looking for. So you can monitor a kind of potentially a much, much larger breadth of pathogens all at once. But there's some trade-offs, of course, and that is that it's less quantitative, it's less sensitive, and often you can waste a lot of information or get a whole bunch of sequences for other stuff that's in your wastewater that are not pathogens. And so there are some challenges with sequencing-based screening as well. But we have a couple of projects where we're working on this. I have one postdoc who basically mined all of the publicly available wastewater sequencing data that's available on NCBI Sequence Read Archive. It's like terabytes worth of data. And we just said, okay, which pathogens can be detected in this massive amount of data that's publicly available? And we're using this to kind of identify potential targets that might be amenable to wastewater screening. We've also actually done a study where we sequenced some of wastewater in Houston. So we took just a snapshot. So 14 sites, we got some wastewater samples. We enriched for viruses out of them, and then we sequenced it. And we were able to detect kind of a really broad suite of viruses in our wastewater samples, from enteric viruses, respiratory, bloodborne, vector-borne, and then some other viruses, like ones transmitted through skin or saliva. And just a little closer look at these. We sampled at wastewater treatment plants, but we also sampled at some of these congregate living facilities and schools. And one, initial takeaway was that, okay, these wastewater treatment plants, this is showing a stacked bar graph of all the different families of vertebrate viruses that we detected. The wastewater treatment plants all look relatively similar to one another, which kind of suggests this averaging effect. So when you sample at the treatment plant, you really are getting this kind of like averaged effect across the population. And the treatment plants look pretty distinct from the manhole samples. So, these all feed into the wastewater treatment plants, but as it gets there, lots of other things are contributing, resulting in this, pretty reliable, maybe robust signal at the treatment plant. But there may be some viruses that are only really detectable in a sensitive fashion when you sample at the facility level. We also looked and tried to ask, okay, do we see any notable viruses in these samples? And we saw, a number of different things like HPV and specifically cancer-causing HPV. We detected HIV, and we also detected several different herpes viruses. And so these all could be potential future targets that we could develop more targeted methods for to actually do quantification and routine surveillance. And that's kind of our next step, is thinking about what should be the next round of routine surveillance. What's the frequency at which we would monitor for these targets? What action could be taken from additional information that wastewater could provide on these different targets? And then this is, I think, my last summary slide. I think there's one thing you should take away. It's that wastewater monitoring for public health is really here to stay. It's been demonstrated at this massive scale. I stole this slide from the CDC, or I stole the text from it. This is kind of their vision, which is our federal agency, the Center for Disease Control, for what wastewater surveillance should be moving forward. They envision a core surveillance program where you do regular surveillance of endemic or common diseases, an emergency response aspect of wastewater surveillance where you use it when you see an outbreak. And then we can also use wastewater surveillance for special studies to kind of get information on specific diseases of concern that might be specific to a region or a specific time point. And with that, I just want to acknowledge there's been so many people who have contributed along the way to this project that have made it a success from many different institutions. None of this work would have been possible without the contributions of all of these individuals. And of course, our funding. So we receive support for this from many different organizations, including Rice. It's primarily supported by the Health Department and CDC funding now. And I also want to acknowledge my amazing lab group at Rice. Almost all of these people have contributed at one point to this program. And I'm happy to take questions.
Let me be the first, Dr. Lauren, to take a few or ask you a few questions, if I may be allowed, Jennifer and Ragashri, Professor.
Please go ahead. Yeah, I have a few questions too. Yeah, go ahead.
There are several questions from several participants as well. Being a layman in this subject, so to say, on wastewater epidemiology, why samples have to be taken at WWTPs and not at the sewage conveyance system is one of my first questions. Okay, the second would be on what is the minimum area of surveillance that the WBE should be applied or should be tried. And the third would be on what is the type and size of data that is generated in India. You have showed a few locations in India where the WBE is slightly giving some data. we heard in the past from Sangeeta Adhikari also, there is something happening in India also. And we have some experts who said something is happening in Bangalore. You showed the map in Bangalore where there is some work happening. So few of these questions, if you may would like to answer.
Great. Yeah. Okay. So the first one was, why do you have to take it at the wastewater treatment plant versus within the sewer system? And you're completely right. You can collect a sample within the sewer system, and you don't have to collect at the treatment plant. We started at the treatment plants because there were existing auto samplers there, because they were collecting these samples already because of routine monitoring of their treatment plants. And so when we were trying to spin this up really fast, in order to get all those samplers that give you the composite samples instead of the grab samples, we had to, the health department had to order hundreds of these and then install them. And so that took time. And so while we waited for new ones to come in that were placed within the sewer shed, we used the existing ones at the treatment plants. But you can certainly monitor within the sewer system. So we monitor at lift stations, which are basically the pump stations within the sewer system, where wastewater is basically pumped up before gravity flows to the treatment plant. But you can also monitor, manholes at individual facilities. And that's what we're doing, for example, for the schools and things like that. What's the smallest size? This is a really, really important question. There's a huge, all these individuals working in wastewater-based epidemiology are thinking about the ethics around this, right? Because it is, also even just the word like surveillance and monitoring, it has like negative connotations to it, and where you have to build public trust for people to embrace this approach. And so as you go to smaller and smaller communities, you come into this point where, okay, you have to question whether or not you're applying this, in a way that's transparent, and that the information can't be traced back to individuals, for example. And so I know people have applied it at the building level. And I think that's the smallest application I've seen of it, or actually at the airplane level. I don't know if you've seen these. People have screened airplane wastewater for SARS-CoV-2 to see if like travelers entering a country from other countries are infected. So it has been applied at a much smaller scale, but as you go smaller and smaller, one, the advantages of like the community pooled sample get diminished, so it's less resource efficient. And then two, you have to start questioning kind of the ethics of it, because you don't want to stigmatize individual communities. And what was the last question about? It was about
what is the type of data
India? That's
India?
That's a great question. I don't actually know. We can click on those and see if there's a publicly facing dashboard. I think there may have been one, but maybe not. But I actually am not sure exactly what's going on in terms of monitoring programs across India. I've heard lots of people talking about it as well, and people trying to spin up larger programs. And I don't know how it's being funded internally, but like that's usually that's
one last question, Dr. Lauren. Which is faster? Do we have the RT-PCR tests conducted at the individual level is faster than the wastewater-based epidemiology surveillance results? Which one is faster in conveyance of the particular information of this type of mutations have happened or not?
Yeah, so I would argue that wastewater is going to be faster almost always, because just think about how many people you have to sample to get a representative picture on an individual basis to get what you would get with a community sample with one grab of wastewater. And even practically speaking, like the way that case data comes in clinically, it's happening at all these different facilities all across our city, has to get aggregated to our, sent to the health department, has to be cleaned up because there's all these replicate tests. Like I might get tested once it's at one pharmacy and go to another pharmacy when it's inconclusive and get another test, and so health department has to aggregate all this information, clean it up and dereplicate it. And then they finally have this clinical data, whereas the wastewater, it's like you get a sample, you can assay it immediately, and you can get this picture of the whole city. It's not going to tell you where an individual outbreak is going on necessarily, unless you have really good spatially resolved samples. Yeah. But it can tell you very early on whether or not you see a change in a viral concentration across the city.
Thank you, Dr. Lauren. Professor, you have your questions.
Well, beautiful talk lots of insights. It's quite clear from your talk that public health surveillance and public health care will be very different in the years to come. And the fact that governments across are looking at this as measures for tomorrow. So this is measures to care for their public tomorrow. This will change, well, on the one side, disease and related care, but I was particularly interested in also how this is going to look at economics, industry, and environment in greater detail. So this is what we are trying to build along with what you are doing, and it's a great thing. And so obviously we need a lot of help from you in this regard. And I hope to have somebody coming over there and spending a little bit of time, if possible.
That would be wonderful.
Time.
And so, I think you're right. I think you can get a lot of information from this that can impact, economics. You could use this type of monitoring to understand evolution in the environment, right? So you could use it to target diseases that impact plants or animals. It can be applied for many, many different settings and applications, depending on what your questions are. So I think this is just the beginning of it, really. I know it's already exploded, but it's just going to get more exciting.
Well, sure. So you touched upon sort of the negative connotation. Well, we start from there. When you start looking at individual specific information. So obviously public at large are interested in large information, large data sets, etc. And everybody is happy about it. But if you want to go closer and closer, there is concern about it. So at some point in time, you might have also seen opposition. I was curious to know whether there have been instances of this kind, and at what level it becomes surveillance, and at what level it becomes an issue of public concern.
Right. That's a really great question. So I bring it up because this is very much a very active conversation in the wastewater surveillance community in terms of what legal and ethical questions should we be thinking about as we're applying this, because people are not, in most cases, individuals are not opting in, right? They don't choose to have their wastewater surveyed. It's just happening in their communities because someone's grabbing a sample of wastewater that they happen to be contributing to. I think what's critical is how this information is, well, the transparency associated about where these samples are collected, right? And how the information is being used. And then this part of that is this message that this information cannot be used to trace back to an individual, right? If I detect SARS-CoV-2 in a sample, I will never know who contributed to that. In a wastewater treatment plant, even in a facility, you can't trace it back to an individual. There is definitely, the other thing people are concerned about is stigmatizing communities. So wastewater surveillance has also been used, for example, for monitoring for illicit drugs. So things like opioids. And it's really been used by the health departments to decide where to send resources, because there's a huge opioid epidemic here as well, and so knowing where this drug use is going on would help health departments send resources to those communities. But you can see how that could be very easily abused, right? Let's say that got into the hands of law enforcement, and now they're using this information to decide where to send more, law enforcement agencies to basically, find criminals or people who are using, people who have addictions, right? Who are using an illicit drug. And that would be an abuse of this information. And if any example of that were to occur, of course, no one would want to be part of a society or a system where they know that their wastewater is being surveyed and used for that purpose. And so I think we have to be really careful about how we apply this and who has access to the information to make sure that it's not going to be abused. So there have been instances of communities who don't want anyone to sequence their wastewater. So they'll say you can detect the amount of SARS-CoV-2 in the sample, but you're not allowed to sequence it. Even though we don't have any way of tracing back who it belongs to, they're just like categorically opposed to it. And so these researchers will basically drop that pursuit. And then I've also, this is very, there's of course like some people who, deep in the internet, there's like also some conspiracy theories about wastewater surveillance that are like very, people are just worried about surveillance in general. And so I do think wastewater surveillance programs have kind of brought some of those conspiracy theories heightened a little bit. And that's something we always have to worry about, right? Because people get so much information on the internet and don't know how to like tell what's real versus what's not really supported by science.
Last two more quick questions and then leave it to others. You touched upon, or I also mentioned that things will be changing tomorrow. And one of the things that environmental surveillance has done, carbon dioxide levels, particulate matter levels and all that has impacted real estate. Economics has been affected. Good and bad in whatever ways. So obviously I see that the way things are moving forward, real estate can be a concern. So if a lot of people are constantly being affected by certain kinds of diseases, people may not live there, right?
Definitely.
Going further, many other related things. So there is an economic side to it. And have you started seeing, or have you seen any kind of thing that in my country, when certain epidemics coming up, nobody wants to go there. Tourism gets affected. Obviously,
that's a great question. I don't know if we've seen that, right? But I don't know if we've like necessarily looked for that, if that makes sense. I don't know if we would even be able to see that on a citywide level. I think you might be able to see that on a maybe even like a larger geographic area. But I think you're right. This is, again, one of the worries of using this approach where you can stigmatize specific communities where you see, okay, this community increasingly has a disease burden that's higher than this other community, which probably has to do with lots of factors, right? There's lots of ways that we could already get this information without doing wastewater surveillance, right? And that could impact the value of homes in those areas, real estate, things like that.
So the last one was that communities or India, we have often wastewater is mixed with stormwater. And wastewater is not wastewater anymore. So therefore, the kind of sample load depends upon monsoons and torrential rains and whatever kind. So do you see incidents of this kind? And in such scenarios, how do you extract the information?
Yeah, this is really an important question. So Houston has separate sewers, so we don't have as much of the dilution effect that occurs when there's a storm because we have separate stormwater from wastewater. That being said, there's tons of inflow and infiltration, and many cities across the US also have this combined sewer system. And so the question is, how should you normalize your data, right? And so there's a few different approaches. We use a flow-based normalization. So that's why we report a viral load. And so we multiply the concentration times the flow rate. And so the idea is if we have a huge storm, your flow rate will get really, really big, which will increase your viral load a lot, but then your concentration will be significantly diluted. So those two will balance each other out. The other thing that people often do is they measure a fecal indicator. So they'll measure something like, a virus that's like very prevalent in all wastewater. So like PMMOV is one of them, the pepper modal something virus, modal virus. No, I can't remember, it's PMMOV. There's also some other viruses that are good fecal indicators like crassphage. And so they'll measure that virus and they'll normalize the SARS-CoV-2 signal to that fecal indicator. I think there's really no, this is the best way to do it. This is one of these ongoing debates, I think, in the community. We are really pro flow normalization because it's worked really well for us. And we've seen that the fecal indicator normalization often introduces a lot more noise because, if there's variability in your fecal indicator, you're dividing your signal by a variable number. So you get even noisier data. But other groups swear by it. So I don't think there's definitely not a one size fits all type solution for this.
Thank you. I see 3 questions here. Lourdes, Xavier, Jennifer, and Chandrasekharan. So you please go ahead, Jennifer. You please run it the way you like. Yeah.
So I have many questions, but I'll limit to a few. Are there any data updated to you from regarding the public movement? Say people are moving out of their places during the weekends. And is it important, or can we just rely on the fecal indicators alone? And my next question is moving to other technologies other than PCR. As you know, there could be some supply chain issues in creating the primer probes, identifying the mutants using the probes. So one of those we were looking at was mass spectrometry. And have you come across any other techniques to identify the viral load? Yeah, these 2 are my questions.
Yeah. So the first question about movement of people is a really important one. And I think, we think about this a lot because, in our 39 wastewater treatment plants, people are probably not living and going to the bathroom in the same sewer shed that they're working and going to the bathroom in. And so you may be contributing to multiple different sewer sheds across the day if you're infected. And so I think that explains some of the noise that we see. And that's often actually why we try and look at it at this aggregate picture and try to say, okay, we'll look at it all together. What are the trends that we see across the city? Because we know people are moving across the city. And like you said, people are moving out or into the city, depending on big tourist events or holidays and things like that. We did look at the most recent big event here in Houston was the rodeo. So we did look at across the rodeo. I'm not sure if I don't have any data to show for that, but we did look across the rodeo to see if there was an impact of that on COVID. I know there's a number of other cities like Las Vegas that gets tons of influx of tourists, and they've spent a lot of time looking at what is the signal coming from the tourist populations versus the residents of Las Vegas, and how do we kind of disentangle those 2 signals? But I think that's going to be really important, especially if you live in a city that's super tourist attractive, especially holidays or festivals that bring in lots of people. Your other question about PCR, I think this is a challenge that we're all dealing with in terms of supply chain. I don't know if there are as robust and sensitive methods for SARS-CoV-2 as PCR-based ones. I have heard of people using mass spectrometry. A group out of Arizona is doing that, I know, Rolf Halden. And I think you can use mass spectrometry to look for viral proteins basically in your sample. I don't know enough about it to know if it's as sensitive. I assume it's less high throughput, but that might be just my, lack of knowledge about the sample preparation, the time for analysis. I think people like PCR because it's relatively high throughput and quite sensitive, right? You can potentially detect single copies of genes because you have this amplification step. With mass spec, there's not the equivalent amplification, right, of the target material. So it's the sensitivity might suffer for that.
Okay, I see other questions. Lourdes was there to begin with, actually, and then Chandrasekharan. Lourdes, are you still there?
Yeah. Okay, I can ask a question. Hi, Lauren. Great talk. This is a question regarding genomic analysis. You have done a lot of genome analysis and studied mutations. My question is, have you tried to use the metagenomic analysis of SARS-CoV-2 from wastewater, leading to direct structure prediction of evolving viral structures, given the recent advances in structure prediction with AlphaFold and RosettaFold? Like, if we can directly predict the protein structure, which is evolving, it would be useful to discover drugs, like directly from wastewater.
Interesting. We have not done that. I think that's an interesting idea to pursue, maybe not specifically even for SARS-CoV-2, but for other rapidly evolving viral genomes. That could be a great area of exploration. It seems like a really cool application of this. Again, you run into this challenge of it being this mixture, right? So sometimes it's hard when you have these tiny fragments to know that they all originated from the same genome. And I think that's one of the biggest challenges we have with this like short read sequencing and then trying to use that information. Because I have like all these short sequences. Did they come from the same original genome, or are they actually present from 10 different genomes that were all in the same sample? So you're always going to run into that problem. We have started to look at analyzing in longer, well, not longer, but the entire reads and try to say, okay, what novel mutations can we detect on these reads that we wouldn't have otherwise detected through clinical sequencing or other publicly available sequencing information. And I think you could potentially use a similar approach, I guess, into looking at rapid evolution of different viruses and viral genomes, and even use that to look at protein structures. But that's definitely outside of my area, but it sounds cool.
Thank you.
All right, Chandrasekharan, you have a question?
No, sir, just an appreciation. She has done a great job, and it was very relevant to the immediate need of the people over there. So I should appreciate and congratulate her. Thank you. And also the question by Paul, that was really insightful. I think our scientists will take the clue and keep working on it in collaboration with your great team. Thank you.
Thank you. I hope they do pursue that. It sounds very interesting.
So, well, Lourdes has been always very, very insightful when it comes to questions. And that is, well, hopefully people will pursue that. Lourdes himself, you will pursue it, I suppose.
Yes, it's a kind of hottest topic now, directly predicting structures from genome and protein sequence.
Right. Yeah. Okay, thank you. Sripati.
Nothing more to add at this point of time, but the only thing which I gather from this topic or this excellent effort that you have been pursuing, Dr. Lauren, is that the scope is very, very huge. The scope of work on this subject. And one pertinent question, you also indicated in the first or second slide, I don't remember, they started thinking about wastewater epidemiology only after COVID, isn't it? Why didn't they think something before that? Is probably a question I would like to know from you, an answer from you on this. Why didn't they?
I think that many groups, but they're more academic groups, though in Europe, they actually did apply it in conjunction with health departments. They were using wastewater surveillance prior to COVID to monitor for other viral diseases and also to monitor for illicit drugs like opioids. And people have also used it to actually even look for nicotine, or nicotine byproducts to look at, smoking use and other indicators of health in communities. I don't think it was really this pandemic, though, that brought it to wide spread adoption and application. And so it had been used by pockets of groups, but never really embraced in the way that it has been since the pandemic. And now I think because it was demonstrated as such value, many health departments who previously didn't realize it's just this entirely additional piece of information that they didn't have, right? They have their traditional surveillance methods, which are basically all based on individual testing. And those are so resource intensive and targeted, whereas wastewater provides this whole additional piece of information that's much broader in terms of the number of people you can capture, less sensitive potentially, right? But provides this new piece of information that didn't previously had, and now everybody sees the value of that.
Probably the last question, since I'm handling the implementation vertical at this center, ICCW, what would be the type of funding that may come to India if at all these kind of studies or these kind of research is going to happen in India? Are there any?
I don't know if I know the answer to that question, to be totally honest. I think, yeah, it's a matter of like who funds this type of work. You know, at the national level, the state level, even the local level. Ours was largely championed by our local health department. You know, it was very much, and now they're getting they get federal funds to support it, but they're choosing to take the money they get from the federal organization and prioritize this because they see the value of it. In India, I'm not sure what the funding structure is that results in kind of a similar support system, but it's definitely, yeah, I don't know. It kind of depends on the, yeah.
Ideas come from this will decide funding, and there is no limit. That's what I see. Yeah.
Thank you, Dr. Lauren, for your time at this early part of the day, and enlightening us on several aspects of the work that you have done. And this brings out or opens a Pandora's box for us to think many things to work upon. And as Professor said, somebody from this side would come to your place and learn a lot of things from you, and try and do something at India also, in several aspects that you have been trying to work on. Professor Pradeep and Jennifer, they have been working on this topic, and they are coming to ICCW to do something with ICCW on this topic. I would suggest that we will have to learn a lot from you, Dr. Lauren, and take this subject forward. And very, very, insightful, data that you have shown. And we have some people in our ICCW working on data analytics as well. We are looking for such data that could develop so many insights into the aspects of the pandemic and the next sort of sets of genomes and the mutations that are going to happen. So it's going to be a huge amount of scope of work that we will be getting into. So thank you once again on behalf of ICCW and IIT Madras to Dr. Lauren. On behalf of Professor Pradeep and Jennifer, we would like to thank Dr. Lauren to you once again, and we look forward to meeting you more times, many more times in the coming days and months to learn a lot from you and try and do something in India in the same topic of wastewater-based epidemiology. Thank you, Professor. Thank you, Dr. Lauren. Thank you, Jennifer.
Thank you.
Chandra, and all the participants from the group. Have a good day. We'll meet again.
Hi, thank you so much.
Thank you. Thank you.